If you want to understand why some people keep fighting cancer into their 80s, while others collapse at 50, don't look at chemotherapy. Don't look at genetics. Look at a tiny black seed that modern medicine quietly stopped talking about. In medical libraries around the world, a quiet surge is unfolding. Over the last 20 years, the number of published studies on Nigella sativa, also called black seed, black cumin, or kalonji, has soared from just a handful each year to more than 2,000 peer-reviewed articles in total. Researchers from leading universities are mapping out thymoquinone's effects on stem cells, cancer pathways, immune signaling, and related biology. In the past decade alone, the pace of publication has more than tripled, with new papers appearing every month in journals of pharmacology, oncology, and regenerative medicine. Yet, as the evidence piles up in laboratory notebooks and specialty journals, the silence in clinical practice is deafening. No major medical society, whether it is the American Cancer Society, the National Comprehensive Cancer Network, or the European Society for Medical Oncology, mentions black seed or thymoquinone in their treatment guidelines. A search of the guidelines' international network database returns 0 results for Nigella sativa under any of its common names. Even in areas where the preclinical data are most promising, such as stem cell biology and anti-inflammatory therapy, not a single national or international guideline recommends its use. Doctors and patients alike remain in the dark. The disconnect is stark, a wave of scientific curiosity on one side and a wall of clinical inertia on the other. For every new study exploring how thymoquinone might triple stem cell counts or starve tumor cells, there is a matching absence of official recognition or practical advice. The question lingers: how can so much research generate so little action? And what keeps this ancient remedy locked out of mainstream medicine, despite a growing body of evidence? The roadblock isn't a lack of evidence, it's a matter of economics and cultural inertia. Natural seeds like Nigella sativa cannot be patented in their original form. Without exclusive rights, pharmaceutical companies have little incentive to invest the hundreds of millions of dollars needed for large-scale clinical trials. The average cost to bring a new drug to market now exceeds $1.3 billion, a figure justified by the promise of intellectual property protection and future profits. For black seed, there is no such financial safety net. Any company that funded a breakthrough study would see their results freely used by competitors, with no way to recoup the investment. This economic reality explains why the bulk of research on black seed and thymoquinone remains limited to small, university-backed studies or government-funded grants. Those projects rarely progress to the kind of definitive trials that change medical guidelines. But money isn't the only barrier. Black seed's reputation is shaped by its long, cross-cultural tradition. Ancient Egyptian physicians prescribed it for digestive complaints as early as 1550 BC, recorded in the Ebers Papyrus. Greco-Roman healers, Islamic scholars, and Ayurvedic practitioners all described its benefits centuries before modern pharmacology existed. For decades, this legacy was dismissed as folklore, not science, a bias that lingers in medical institutions today. Even as new studies appear in respected journals, the weight of tradition can make it harder for these findings to break through the skepticism of regulatory committees and clinical guideline panels. The result is a system where promising natural compounds are often sidelined, not because the evidence is weak, but because the machinery that drives medical progress is built to reward ownership and novelty, not rediscovery. In this climate, ancient remedies like black seed remain trapped at the margins, their potential recognized in the lab but rarely translated into everyday clinical advice. Nigella sativa, known in many households as black seed or kalonji, owes its reputation to a single molecule, thymoquinone. Laboratory researchers have spent years testing this compound's effects on the body's repair system, hematopoietic stem cells. In a controlled animal study, mice given black seed oil showed a striking change. The percentage of bone marrow cells marked as LSK, the gold standard signature for stem cells, rose from 0.23% in untreated animals to 0.71% in those given the oil. That is a 209% increase, sometimes rounded in popular accounts to tripling. In parallel, colony-forming unit assays revealed a 3.1-fold rise in the number of cells able to grow into new blood and immune cells. Flow cytometry and cell culture confirmed these results, but so far, all of this evidence comes from animal models, not people. The findings are real, but the leap from mouse marrow to human medicine is still underway. Even so, these numbers have turned a centuries-old folk remedy into a measurable biological effect. Researchers are now racing to explain it at the molecular level. Thymoquinone's reputation as a multi-target molecule comes from its ability to interrupt cancer biology on several fronts. In one set of experiments, human endothelial cells exposed to 10 micromolar thymoquinone showed a 45% drop in tube formation, a lab stand-in for blood vessel growth. This effect traces back to VEGF, the signal protein tumors use to recruit their own blood supply. When VEGF is suppressed, tumors are cut off from nutrients and oxygen, stalling their expansion. But thymoquinone doesn't stop at starving tumors. In cancer cell lines, it triggers a powerful wave of programmed cell death called apoptosis. Researchers measure this by tracking caspase-3, a molecular executioner that dismantles cells from within. Caspase-3 activation climbs sharply after thymoquinone treatment, while markers of cell survival decline. The third mechanism targets inflammation at its roots. Thymoquinone stabilizes IKBA, a protein that keeps the inflammatory master switch, NFKB, turned off. In animal models, this means up to a 40% reduction in NFKB's ability to bind DNA and drive the production of cancer-promoting signals. Each pathway, angiogenesis, apoptosis, and inflammation, represents a separate axis of disease. Yet thymoquinone intervenes at all three, suggesting a breadth of action rarely seen in a single natural compound. Dr. Carter stands before a wall of scientific journals, the numbers speaking for themselves. More than 2,000 papers now mention Nigella sativa and thymoquinone, a surge that has turned this ancient remedy into a modern research obsession. But when you look closer, most of these studies never leave the laboratory bench. Less than 10% involve actual people. Out of over 2,000 publications, fewer than a dozen are controlled human trials. The rest, cell cultures, animal models, and mechanistic reviews, build a case for potential, but stop short of real-world proof. Meta-analyses and systematic reviews echo the same pattern, promising signals, but not enough human data to clear the bar for clinical confidence. There is no shortage of mechanistic evidence, but the leap from petri dish to patient remains incomplete. Until large-scale trials fill this gap, the conversation around black seed stays rooted in possibility, not prescription. This is why practical protocols still require careful translation, and why evidence badges in this field come with a disclaimer. Exciting, but not yet definitive. Dr. Carter lays out the protocol step by step, careful to match research with real-world practice. For those reaching for black seed oil, the most studied dose in human trials is 1 teaspoon, about 5 milliliters, taken twice daily with food. This routine lines up with the majority of published studies, where participants used between 1 to 3 grams of oil per day for 8 to 12 weeks. For those who prefer the seeds themselves, 1 gram daily of lightly crushed black seed can be sprinkled onto breakfast or blended into smoothies. Capsules offer another option, 500 milligrams standardized to at least 1% thymoquinone, taken twice a day. Some people combine half a teaspoon of oil with a spoonful of raw honey first thing in the morning, a practice with roots in traditional medicine and still used in clinical research for taste and absorption. Cooking with black seed oil 3 to 5 times a week, especially alongside cruciferous vegetables like broccoli, may help integrate the protocol into everyday meals. Each of these steps is designed for consistency, not intensity. The goal is a sustainable habit, one that can be maintained for at least 90 days, giving the body time to respond. No single format is mandatory. Oil, seeds, capsules, and culinary use can be mixed based on preference and tolerance. The regimen is practical, evidence-aligned, and flexible enough to fit most daily routines. Cold-pressed black seed oil stays best when refrigerated, sealed tightly in dark glass to protect thymoquinone from heat and light. Capsules should list at least 1% thymoquinone and provide a certificate of analysis from a trusted lab. Whole seeds belong in a cool, dry spot and should be crushed only right before use to preserve their natural oils. Studies show that daily use for 8 to 16 weeks, never less than 90 days, is needed for meaningful results. Stopping early or skipping days can weaken the benefits for immune and stem cell function. Black seed oil and thymoquinone are not safe. During pregnancy, breastfeeding, or for those planning to conceive, due to limited safety data and risks seen in animal studies. People on blood thinners such as warfarin or direct oral anticoagulants should avoid black seed because it may increase bleeding. Those with low blood pressure or people taking antihypertensives need to watch for further drops. Anyone actively receiving chemotherapy or radiation must consult their oncologist first, since interactions remain unclear. Product quality varies. Independent testing often finds thymoquinone levels lower than labels claim. Third-party verification is essential for safety and efficacy. Every day, new studies reveal how overlooked molecules like thymoquinone reshape our understanding of biology, with no patents required. The future of cancer prevention won't come from magic, but from evidence and curiosity. The real question: who decides which discoveries reach us? Your health deserves more than silence. Share your thoughts below.